Hello and welcome to an addendum to Odds and Ends number 10. In this episode, I'll talk about some experimental lines regarding nitrogen fixation within grain crops such as wheat. Researchers such as Dr. Ivan R. Kennedy have been working with various agricultural chemistry companies to develop a means to produce nitrogen through biological means in hopes of reducing the amount of synthetic fertilizer being used in agriculture. Now I must bear in mind, most of this stuff is experimental and will likely not be out as a practical option for quite some time. For context, legumes naturally produce nodules that allow for a symbiotic relationship between a nitrogen-fixing bacteria called Rhizobia bacteria, which includes multiple different genera, and the plant itself. Typically, each plant has its own specific Rhizobium bacteria that forms a relationship with each species having differing levels of nitrogen fixation capabilities. Wheat crops and most other crops in general do not have the capability of producing nodules. That's a phenomenon found only in legumes. So various other experiments were performed to see if you can simulate this with other methods and other bacteria or microbe species. One such attempt was a work by Dr. Kennedy and Dr. Chan, which attempted to allow for soil colonization of Datotrophs, a soil microbe that fixes nitrogen into the soil, and are often found at the rhizoplane or root surface of various cereal species. It didn't work because the roots release insufficient amount of carbon for the Datotrophs to colonize that region, and there's heavy competition from other microbial species. There's also hopes for finding Datotrophs that can be grown within the root system, but it's unknown if that's possible. There's also the problem of the oxygen paradox. Most of these microbes require oxygen, but the more oxygen they have, the less nitrogen they produce. And finally, the transfer of nitrogen to the host plant is much less efficient than legume crops or with crops with applied nitrogen. Recent bits of research suggest that different species of nitrogen-fixing bacteria, such as an endophyte bacteria called Actobacter dalophysis, could colonize sugarcane if inoculated during propagation. This research, developed in the 80s, suggested that multiple of nitrogen-fixing bacteria could colonize the root systems, allowing for a high degree of nitrogen fixation, although, once again, this is still experimental. Another species that was tested was extracted from caloe grass. These are Azoria species. These species are able to commingle with its host plant and produce nitrogen. Another symbiotic microbe would be Hyphotrophes, which allowed for the grass plant, specifically rice, to produce nodule-like structures. Although whether or not you can call them nodules is dubious, evidence of colonization and nitrogen fixation has been observed. Now on to wheat. An interesting species of bacteria called Parasponia and its relationship with wheat has been studied by Dr. Cochrane in 1992 and found these two species tend to fuse together, creating infections associated with shortened, thickened lateral roots, indicating a more primitive form of Rhizobia. His associate, Dr. Chan et al., also in 1992, studied Azorhizobia in cereals as well. This researcher found that varying levels of symbiotic relationship and bacterial colonization happened depending on the species. It was also found that a chemical additive, nitrogenin, stimulated colonization of the wheat roots by this bacteria. Like with the symbiotic relationship with sugarcane bacteria and sugarcane itself, most of this nitrogen fixation colonization happened in the lateral roots, and the amount of nitrogen produced by the endophyte species was akin to the primitive Protorhizobium, far higher than Azoparium brasiliensis, although there is less colonization within the A. canadensis species. Other researchers used 2,4-D to initiate colonization as well, as an experimental plan. Although it's impractical as a management strategy due to labor and chemical costs, it became a proof of concept to try to create new mutants of different species of symbiotic bacteria for future nitrogen production. This also led to the discovery of several genes that promote nitrogen fixation, such as the NIF genes, the LAC-Z genes in the Astrospirilla species, as well as a recent study that showcased the appearance of the GS-A gene. These were discovered in the late 90s. Within Australia, a mutated form of Aspergillus brasiliensis called SP5 was developed. This mutant had reduced EPS capabilities, which is the ability to produce exopolysaccharides. This prevented the bacteria from adhering to the root system, but also increased the rate of colonization. This increased the amount of nodules or pseudonodules being formed within the wheat plants in various studies. A variant on SP7 called SP7S was further refined to develop a mutant that feeds more so on glucose than other plant-based chemicals. This improved the growth rate within the root colony, but it lacked the ability to produce nitrogen. The final version, as of the writing of this paper, was a double mutant of SP7 called SP7SA. This mutant was a hyperproducer of ammonium, but this also came with the problem of inhibited colonization, as this double mutant could not colonize as well as the previous two variants. The current work in Australia is to develop strains of this bacteria that can colonize the grain as well as produce sufficient amounts of nitrogen and function within the root system far better than it currently can. It'll take a lot of time and effort for that to happen, but hopefully it'll someday be successful. This paper showcases the great difficulty of developing new technologies and new management strategies for agriculture. Sometimes research and development can take decades, and it's often built upon partial victories and failures and groundwork of papers that came before us. Such is the nature of science, and that about covers everything. Thank you for watching.